Yes. So again, switching to a different topic here, and that's something that I tend to call spatially aware methods. And with that, I mean that here we try to actually include the spatial structure of your data in the analysis, and not only to visualize or map down the results to you. When we looked at this breast cancer example, you could see that, sure, we visualized the clusters spatially, but the clustering process itself had no notion of spatial positions of our data points. And this is sort of like what you try to then now include in your analysis. And some examples of what you can do here is to identify spatially variable genes. And I will speak a bit more soon about why we are interested in spatially variable genes and not just highly variable genes. You can also do this to find spatially coherent expression domains. You can try to leverage spatial proximity to increase robustness of your inference, such as CNA inference, or to find local correlation between features. Let's see here. Yes. So speaking of these spatially variable genes, then, the main idea here is to somehow either binary sort your genes, saying these genes have a clear spatial patterns, these genes do not have a clear spatial patterns, or perhaps put them along a gradient, saying these genes have a strong spatial pattern, these have a weaker spatial pattern, but to somehow find a way to automatically, because it's not super nice to browse through 20,000 different genes by eye, to sort them along this gradient or make a binary classification. And methods like Spatial DE, SVCA, and Spark, they tend to use these probabilistic models, and they all more or less to some extent leverage these Gaussian processes, which are not really the same thing as a multivariate Gaussian. Sometimes that's mixed up, but they are related, of course, to model the data. And then they, again, essentially test whether a spatial term in the covariance function significantly increases the model's ability to explain the data. And if it does, well, we have a spatial pattern. And if it doesn't, it's not a significantly spatially variable gene. A slightly different approach that I took here in a method that we've been working on called Spatial is not at all probabilistic, which is sort of funny because that's usually my way modus operandi. But here we use a very deterministic approach. We use finite differences, which is a numerical method to simulate diffusion of transcripts within the tissue using fixed second law of diffusion. And the idea here is then measure the time until you get a converged state. So if you start with a fairly structured state, you let all these transcripts diffuse in the tissue, and when this homogeneous state is reached, you stop the timer. And then you can rank all the genes by the time it takes to converge. And the idea here is that the longer the time it takes, the more structured the initial state was to begin with. An already very random state would reach a random state pretty fast. And now to touch upon why we are interested in spatially variable genes. And what you see here is a bit of a supplementary to this Spatial publication, which we had to add because the viewers are very keen on seeing this. So what you see here are 20 expression profiles from the mouse brain. And in the top row, you have, in the top and third row, I should say, you have the real observed expression profiles. And in the second and fourth row, we have the very same data, but where we shuffle the spatial locations. So same values, different spatial location. Now, if you were to just compute variance or dispersion metrics for this whole data set, the shuffled and non-shuffled profiles would end up with the exact same metrics, since these sort of like computation doesn't really take spatial structure into consideration. However, if you apply a method like Spatial, Spatially, Spark, or any other one of these like methods devised to find spatially variable genes, they tend to rank or separate the actual true expression profiles from the shuffled ones, and that's sort of like what we are after here. But that sort of like shows why it's nice to have the spatial component included in your analysis if your data is spatial. A different way that you can include spatial information is to extract, like I said, spatially coherent domains or patterns or clusters. And like we spoke of before, normal clustering mainly focuses on the gene expression. You want data points that reside near each other in gene expression space to be in the same cluster. So sort of like this scenario here, where we have like a nice distinction between our clusters is favored, while this mixed distinction of our clusters, mixed like setup of our clusters is sort of like disencouraged. That's not really what a good clustering algorithm would produce. It would favor this to the left. However, we can also try to leverage the spatial information to find more spatially coherent clusters. And a very common way of doing this is to use something that's called hidden marker random fields, HMRF. And here you construct a graph based on spatial proximity. So cells that reside near each other in physical space are linked together in this graph. And then you say that the probability of a node or like cell or spot or whatever your like spatial locations are belonging to a specific domain depends on two things. Partially the same thing as before, you want proximity in gene expression space, but you also want coherence with your neighbors in the spatial real physical space. So it would be favored to have, if this represents, for example, Visium spots, that all these neighboring spots are of this belong to the same cluster, while having this red spot here in the middle of 4 blue spots wouldn't be really encouraged. And you try then to weigh these two criteria against each other to somehow extract sort of spatially coherent domains. And you can find an example of this in the publication I referenced below here. One tool that is using this approach is called STARCH, and they try to infer copy number aberrations from spatial transcriptomics data. And the idea here is that they want to increase the robustness of this inference by aggregating data in the same domains, since they posit that they should have sort of similar profiles. That is currently in bioRxiv, but it seems to be like giving fairly good results. A sort of different method here that doesn't really focus on finding coherent domains, but rather local correlations is SCHOT, where you compute these spatially weighted correlations. And the idea here is that perhaps computing a single correlation value for genes across your tissue isn't really the best way to go. Maybe you want to look at like smaller regions and see if you have correlations that are local. And as an example, they give 2 genes here, ARRB1 and mTOR, which to the naked eye doesn't really seem very exciting or anything special. Or to me, it just looks like a mess. But apparently then when you apply their method here, you can see how it seems to show some sort of anti-correlation here in the granule layer and more of a like positive correlation in the glomerular layer, I think it's called. Like I said, not a biologist, so yeah, take it for what it is. Then another sort of like topic or field is deep learning, and I put that as a category even though it's extremely broad, and it could also like be interwoven into the other topics or fields here. The reason for saying like putting this sort of like as its own field is because it's very nascent and we have relatively few examples of it in spatial transcriptomics, and that is mainly due to a limited amount of high quality available data. We know that we tend to require quite a lot of data to create good deep learning algorithms. So more traditional machine learning methods has actually been more appropriate to use so far. This is, however, changing as more people produce more and more data and so forth. We, however, have some examples here, being STNet, where we try to relate gene expression to data morphology, and also XFuse, which offers super resolution or pixel resolution of gene expression by learning a joint representation of image and expression data. And I wanted to show a bit of an example here from this XFuse paper by
You're breaking up again.
And here's
Alma,
like structure.
Can you hear me?
Yes.
It seems like when you
Yes,
switch
yeah.
slide and there's heavy slides, then your
Okay.
your network takes it up. Because now when you switched again, it disappears. It always glitches a bit just when you switch slides.
Okay, then I'll pause a bit between every switch. But that's super good to know. Thank you, and sorry for the inconvenience. Yeah, maybe I can just take what I said again. In the leftmost picture A here, you have this unit-like model which tries to learn this joint representation of both histology and expression data. And to the right, you sort of like in the top row see the actual raw observed data that we get from the experiments, while the middle row here shows the inferred expression values at a very much higher resolution. And of course, this could just be humbug and just like be a result. You don't really know if it makes sense. But when you reference it to the LM Brain Atlas in situ hybridization images, it do seem to check out, at least for some genes, which is pretty exciting, a way to combine both images and expression data to gain more from your data.